The stimulation of the alcohol dehydrogenase gene in Drosophila melanogaster, fruit fly, using optogenetics, by Tressa Coultard. Background. Optogenetics is an emerging field that combines optics and genetics to control well-defined events within specific cells of living tissues. It was developed by Carl de and his colleagues at Stanford University in 2006. New technology was created for use in optogenetics, such as to deliver the light deep into organisms, to insert and target light sensitivity to cells of interest, and to assess the effects of this optical control. The genes researchers use for light responsiveness are called opsins. One type of opsins is called channel rhodopsins. The first known and described CHR, channel rhodopsin 1, was identified as a light-gated ion channel in a green unicellular algae from temperate freshwater environments. A second CHR, channel rhodopsin 2, was later characterized from the same organism. Both CHRs respond to blue light and activate neurons, which they are connected to, and increase gene expression, allowing the researcher to stimulate the cells at will. Optogenetics holds promise for clinical treatments with potential for modulating the activity of brain circuits involved in neurological disorders. A method to insert CHR2 into a cell is called Gal4-UAS. The Gal4-UAS system is a biochemical method used to study gene expression and function in organisms. It was developed by Andrea Brand and Norbert Perrimon in 1993, and is considered a powerful technique for studying the expression of genes. The system has two parts: the Gal4 gene, encoding the yeast transcription activator protein Cal4, and the UAS upstream activation sequence, an enhancer to which Gal4 specifically binds to activate gene transcription. Gal4 and UAS are very useful for studying gene expression in Drosophila, as they are not normally present and their expression does not interfere with other processes in the cell. What makes optogenetics exciting to the scientific community is that scientists will now be able to control specific actions, control over defined events with the defined cells types at defined times. Optogenetics delivers a level of precision that is crucial to the understanding of the biological world, even beyond neuroscience. Alcohol Dehyrdogenase Gene, ADH, is a gene that occurs in many organisms and enables the breakdown of alcohol that is otherwise toxic in a yeast and other bacteria. The ADH catalyzes the opposite reaction as part of fermentation. The alcohol is converted to acetaldehyde, a molecule more toxic, which is then quickly converted into citric acid, which is finally converted into energy and used by the surrounding cells. Hypothesis. During the process of optogenetics, when the ADH gene is activated by light, it begins to break down the alcohol at once, whereas when the gene is activated due to the alcohol entering the system, it has to wait until the alcohol begins to be digested. The question is the same amount of RNA produced when activating a gene artificially using optogenetics? In a paper on optogenetics, they mentioned that when stimulating the mice with light, there was increased freezing. Also, they mentioned in description of channel rhodopsins that they increase gene expression. Materials and methods. To determine the amount of RNA produced by the Drosophila require 4 steps. To cross the Gal4 infected Drosophila with the UAS infected Drosophila, to give them alcohol to activate their genes, and in some cases stimulate ADH with optogenetics using a specially made light delivery machine from backyard brains, to extract the RNA out with the RNase kit, and to measure the amount of RNA produced using the real-time polymerase chain reaction machine, RT-PCR. The Drosophila used for this research is Drosophila melanogaster, where they have long been used to study the effects of alcohol, where they have evolved a certain resistance to the effects of alcohol and have acquired the ADH gene. There are 3 groups of Drosophila: Gal4 infected Drosophila, UAS infected Drosophila, and cross Gal4 UAS expressed Drosophila. The 2 groups of either Gal4 or UAS are controls. The Gal4 and UAS do not affect the amount of RNA produced when the ADH gene is activated. Before the Gal4 and UAS infected Drosophila were placed in their vial of fly food, they were exposed to a blue LED light to stimulate their gene. The Drosophila in all three groups were then exposed to food containing 10% alcohol for a 24-hour period. Once the 24 hours passed, the Drosophila in each vial were administered fly nap to put them to sleep, and then carefully, making sure not to mix the groups, put into a mortar and then were killed by a pestle. The tissue from each group was then put into their own conical and covered with RNA later, to store the tissues and to protect the RNA in the tissue. After the tissue was extracted, the RNA was purified from the animal cells, using an RNase mini kit was purchased from QIAGEN. The RNase mini kit purifies the RNA from animal cells, the process of collecting the RNA and washing the tissue. After purification, the final step was to measure the amount of RNA produced. To do this, an RT-PCR machine was used to quantify the RNA and to measure it. Also, an alcohol dehydrogenase gene expression assay was used to amplify the RNA from the alcohol dehydrogenase gene. The data collected from the quantifying and measurement of the RNA was then collected onto the laptop connected to the RT-PCR machine. Analysis. The lab portion of this research is still in progress and results are still pending. The purpose of this research was to test the theory that stimulating the alcohol dehydrogenase gene using optogenetics in the Gal4 UAS system would produce more RNA than when stimulating this gene naturally in Drosophila melanogaster. It was hypothesized that when stimulating the ADH gene through the UAS-controlled CHR2 light channel, more RNA would be produced. Results are still pending and will be analyzed in the near future. Acknowledgements. I would like to acknowledge my teacher Henry Weeks for helping me throughout my project and providing this class so I had the opportunity to have a project. Greg Gage and Timothy Marzulu for custom building my light delivering machine. Stefan Pulver for helping me figure out my hypothesis and finding a company to order genetically engineered fruit flies, and my friends and family who have supported me in my efforts. Thank you.